Hello, I am Jason Lloyd Price, and I will describe our work, Predictability and Randomness of Paw Choices are Critical Elements in the Behavioral Plasticity of Mouse Paw Preference, recently published in Animal Behavior. We are studying how animals acquire biases in handedness when reaching for objects. For this, we place mice into unbiased testing chambers with a feeding tube in the center, so that it takes the same amount of effort to reach for food with either paw. We then record the sequence of paw choices in 50 reaches when individual mice reach for food. In the top graph, we had previously assessed the number of right paw choices that mice make in a right-biased world after they had been trained 1 week earlier with varying number of reaches in a left-biased world. The more the mice are trained in a left-biased world, the weaker is their bias towards the right paw usage in a right-biased world. We concluded that individual biases in paw preference are learned behaviors, which arise even in the absence of any punishment. The distributions show the number of mice that use their right paw a certain number of times in a set of 50 reaches in unbiased testing chambers. The top left histogram was obtained from simulated null model mice which make unbiased random paw choices and thus do not have heavy biases. The other three distributions are from 3 different mouse strains. From each strain we see that genetically identical mice behave differently, and thus this behavior is not constitutive. Further, different strains produce different reproducible distributions, suggesting that a genetic feature affects their behavior. We thus hypothesize that it is the amount of learning from training that differs between mouse strains. In 2 subsequent papers, we used autocorrelation to show that paw preferences change during a training session. This implies that mice have short-term memory of previous choices. First, in the upper left plot, autocorrelations decrease with lag, except for null model mice. The inclinations differ between strains, and therefore they differ in short-term memory skills. Second, in the upper right plot, the 9XCA and BTBR strains, with absent corpus callosum and reduced hippocampal commissure, behave similar to null model mice. This lack of behavior modification for 9XCA and BTBR is visible in the improvement ratio, or IR, seen in the table below, which is defined as the fraction of mice that preferred one paw even more in a second training session in the testing chamber 1 week after the first training session. Note how other strains exhibit high but different improvement ratios, denoting different long-term memory skills. We concluded that strains differ in how much they learn with training, both within and between training sessions, because they differ in short-term and long-term memory skills. So we've seen that mice learn a paw preference from training. However, it's unclear how much information from previous reaches is used in their paw choices. We addressed this question in the present study. We applied an information theoretic measure, the uncertainty coefficient, abbreviated UN, which measures the fraction of information about a mouse's next reach that can be obtained from the bias in the n previous reaches. An example of the uncertainty coefficient from 100 mice of the strain C3H is shown on the right. Note that the immediately preceding choices provide a lot of information about the next choice. After a few reaches, however, the uncertainty coefficient no longer increases with lag, because choices farther in the past lose their influence. Finally, note that UN always stays well below the maximum possible value of 1, showing that, despite the continuous training, there is a component of paw preference that remains random and unpredictable. Comparing different strains, including the null model mice, we see that both the maximum uncertainty coefficient and the number of reaches needed to achieve it vary widely between the strains. However, some things are universal. First, all mice, including BTBR and 9XCA, learn during a training session. Second, for all strains, the number of reaches influencing the next choice is limited, with the strongest learners using only their last 3 to 4 reaches. Lastly, for all strains, there is always a point where the uncertainty coefficient levels out, well below the maximum possible value of 1. In other words, all strains retain a random and unpredictable component in their choices. Finally, note that those strains learning more in a session, that is exhibiting a higher uncertainty coefficient, also exhibit the widest behavior modifications between sessions, as quantified by the improvement ratio. Long-term memory also affects the uncertainty coefficient. First, all mice except 9XCA increase the predictability of their reaches in the second training session. Second, except for BTBR and 9XCA, all mice achieved their maximum predictability with fewer reaches in the second session. Third, although predictability increased with training, all mouse strains, including the strong learners, retain some randomness and uncertainty in their paw choices. In conclusion, mice use short-term and long-term memory of past events to drive their paw-reaching behavior. This is, by definition, an adaptive behavior based on learning from experience. The efficiency of the learning process is genetically determined. The strain dependence of this efficiency suggests that the number of genes involved may be small. The success of the paw-reaching behavior relies on the use of limited short-term and long-term memory of past events, which is sufficient both in stable environments where any bias is constantly reinforced, as well as in fluctuating environments where constant adaptability, that is the testing of new possibilities, is necessary. Our future work is, among others, to identify the genetic elements of memory acquisition regulation in mouse paw preference behavior, and to understand how this information is stored and processed by the brain. Thank you for listening.